afternoon, everybody. My name is Chris Paxson, and I'm the president of Brown. And my job here this afternoon is just to welcome you to what I think will be a really terrific lecture. This is the first lecture of this year's presidential colloquium series called Thinking Out Loud, Deciphering Mysteries of Our World and Beyond. Sounds a little bit like a movie or something. For those of you who are new to this series, which has been terrific, I think we're in our second year or fourth? 4 years. We're fourth years. How time flies. It's meant to probe enigmatic, important questions in sciences that tantalize us with the possibilities and inspire us, and I hope inspire many students to think about answers and think about how they might contribute to these really important fields. So today, we're just thrilled to have with us Dr. Erich Jarvis. He is professor and head of the Laboratory of Neurogenetics of Language at the Rockefeller University, an investigator at the Howard Hughes Medical Institute. And as we will see, Erich is someone for whom science not only yields answers, but offers a wonderful mix of creativity and collaboration and inclusion. In seeking to understand how the brain learns vocalization, Dr. Jarvis has built a research program that uses songbirds as a model to examine the molecular and genetic mechanisms of vocalization, which is a really complex function, apparently. And it's this evolutionary dimension of vocalization that has captured his imagination for a long time, prompting him to ask why humans are more able than other species to have spoken language. His work is zeroed in on what are called axon guidance molecules, which he hypothesizes are the key to vocal learning because they guide the creation of a crucial neurocircuit in the generation of language. Now, this is not the only thing he's done over his life. He's going to tell us a little bit about his life story, which I think is terrific. But one thing I know about him is that he credits his artistic background, his family of singers and musicians, and his own training as a ballet dancer as a vital source of his interest in understanding complexity and in thinking in really big and interesting ways about what he might do in life to make the world a better place. And so it's no surprise that as an undergraduate at Hunter College, he did something which a lot of Brown students do, which is mix and match different things. He majored in both biology and in mathematics because he couldn't decide whether to study, and I quote him, how the brain worked or the origins of the universe, one or the other. He is as well a passionate advocate for science and for greater diversity in scientific fields, believing, as I think many of us would agree with, that diversity brings different views and different ways of thinking about the world in ways that strengthen scientific discovery. In his stirring speech at the March for Science rally in Washington, D.C. on Earth Day this past April, he pointed out that it was congressionally funded NIH diversity programs that gave him the opportunity to become a scientist. And again, I quote, the opportunity to discover how the brain understands and produces the very words he delivered that day. And for good measure, he called for a, quote, science of the people, by the people, and for the people, which I love, noting that the fundamental principles of science transcend political, ethnic, gender, religious, and national boundaries, something we're thinking a lot about recently. So it's this passion and intellectual curiosity and creative spirit that lies at the heart of his lecture today, which is called Dissecting Molecular Mechanisms of Vocal Learning and Spoken Language, A Personal Journey. So I'm grateful that he's here, and I want to turn the program over to Professor Chris Rose, if that's my phone. I'm going to be really embarrassed. Okay, who will introduce Dr. Erich Jarvis. Come on, Chris. I think it was mine.
Thank you, Chris. So whoever got the dirty look, I'm sorry. Okay, so first off, how many recidivists do we have here? So I know Kyle, clearly. Kyle's here. Okay, and that's it? That's the only number of people that have been here before? So this is great. Oh, no, it's not great. But no, there's a large number of you who have never been at one of these talks. So when we started this series, 4 years ago, that's kind of scary. The basic idea was to bring in brilliant scientists, aspirational level scientists, folks that us as faculty would like to be. You hear about that all the time. And have them tell us something kind of interesting. Now, there's lots of people that are really good at what they do, but the type of people that we try to bring in for this series are, as I'll say again, as I always say, just a little crazy. And what that means is that they think about the world differently than other people, and they choose problems in ways that are different. So what's interesting is that as this series has evolved, we brought in these wonderful minds. We've had some fantastic talks. By the way, they're all on the web, so you can go take a look at past speakers as well. But one thing that, I guess has been kind of obvious, and that I think in today's climate maybe bears some underlining, is that each and every speaker has been a member of a HUG group, as we now call them at Brown, historically underrepresented group, be it women, Latinx, African American, or Native American. So that's maybe particularly important. And I mention that this year only because that we're sitting in this particular climate. But in addition, the 4 speakers that we're going to have this year, all independently, have included that as part of the story of what they want to tell. And it was unbidden by me. I didn't say anything. I didn't suggest that you should talk about how being who you are informed your particular brand of creativity. So with that, I've already gone on too long. But I'd like to introduce Erich Jarvis, who is, has a certain sort of crazy. And, we're paisans. We grew up in New York, a little bit different. He's a baby. I'm an old man. And, bring him up. And he's done this really fantastic work that he's going to tell us about. And it's about essentially learning how you learn to sing your song, which he has done so admirably during his career and will continue to do. So, Erich?
All right. Thank you very much. Thanks. I want to thank the president, Chris, Andrew, others for inviting me here and to having the confidence in me getting up here to present to you a talk that stands at the bar level that you just set for me. So, and also to see some colleagues like Julie Coward here, who was a colleague when I first arrived at Duke University a while ago. So, so my passion in science is really understanding how the brain works. I decided on the brain as opposed to the origins of the universe because it's connected here on Earth. It's something I can touch, and it controls dancing. And I had a past, and in a way also current pastime as a dancer. Was going to go into that professionally. So, but the brain does a lot of things. And so in trying to pick out what kind of question I would try to focus on, what I learned in the arts is if you play a difficult guitar or difficult piano, then the easy one you play it really well. So I thought, okay, if I pick a difficult problem in neuroscience, then the rest of how the brain works would fall into place. And so I picked something that I thought was complex, and that was vocal learning, which is a substrate for spoken language. I didn't go consciousness. I thought that was too way out there for now. But not heart rate or something like that. So in this particular presentation, I'm going to talk also not only just about the science that we have done over a number of years, but also the personal journey that I have experienced getting there. So the first part will be about the science, and the second part will be about that personal journey, both as a dancer and also as an underrepresented minority. So I broke it down into 3 different topics here, or levels: behavioral, anatomical, and genetic. And really starting with the behavioral, most people know that what we call language, or spoken language, is either unique to us or quite rare. It really depends on this particular behavior called vocal production learning. Only a few groups of animals have it. 5 groups of mammals, us humans of course, cetaceans, these are whales and dolphins, bats, elephants, sea lions, and 3 groups of birds, parrots, songbirds, and hummingbirds. And even the word parrot in many of our different languages becomes synonymous with imitation or mimicry. All of these lineages have close relatives that don't learn how to imitate sounds, like chimpanzees for humans, or subassine birds, we call them, for songbirds. So it's thought that all of these folks here evolved this ability independently, in parallel, from a common ancestor, like the evolution of wings in birds and wings in bats. But we humans experience language as a unitary trait But it does have components that are not necessarily unique to these groups or unique to us that you must understand to understand this topic. One of them is auditory learning. We depend on auditory learning for spoken language, but it's not sufficient. For example, your common pet animals, like dogs, can learn the word sit in English, so siéntese in Spanish, so suwari in Japanese. Or even roll over boy, come get the ball. That's through auditory learning. It understands those sounds. The dog wasn't born with the ability to understand human speech, for example, right? But a dog cannot say, okay, you got it, I will sit, or tell you to sit, okay? But vocal learners can do that, right? Once vocal learning evolves, though, it does come along with a suite of other traits associated with it. One of them is that we depend upon auditory feedback not only to imitate the sounds that we hear, but also to maintain them, such that when we or a songbird or a parrot becomes deaf, we lose our learned vocalizations through what's called deafness-induced vocal deterioration. And without therapy, you can't repair it. This doesn't happen to a non-human primate. We have vocal learning critical periods that we learn best how to imitate sounds and learn a vocal repertoire early in life before puberty that becomes difficult to learn another language later on in life. All vocal learners go through this. And such that there's certain disorders that disrupt critical periods that affect vocal communication more, like autism. That only happens in us. We transmit our languages, our dialects, culturally from one generation to the next, such that when you take a vocal learning species and remove it from its own species, it loses that language. And you get lost of that repertoire due to social isolation. Again, this doesn't happen to non-human primates. Now, not all those, doesn't mean that this is the same as humans and so forth, but there is some structure here or some traits that come along. The other is that once a vocal learner has this ability, it produces learned sounds in a hierarchical fashion. So for humans, we call it speech or song. Even singing is a learned vocalization, spoken language. For songbirds, we just have one word, song. Even when parrots imitate human speech, we just call it, a song-like bird. But you can see that these learned sounds can be broken down into sentences for us, or we call it bouts in the songbird world, or words, or motifs of individual syllables of phonemes themselves. It doesn't mean that these are homologous behaviors. We think they're convergent, but it's evolved in a similar way. Now, we are the most advanced vocal learners, but there doesn't mean that once you have this ability that all the other vocal learners do it equally. There's some more advanced and some more simple. Parrots happen to be among the more advanced non-human vocal learners. And here is an example. This is Disco, a parrot, or a parakeet, as meaning small parrot, who's been raised with humans for 4 years, and this is what he can do.
What seems to be the problem, officer? I am not a cook. My name is Disco. I'm a parakeet.
I'll play that again.
What seems to be the problem,
Just in case you
officer?
don't believe
I
it.
am not a cook. My name is Disco. I'm a parakeet.
So Disco is a parakeet, and obviously he says he is. And I mean, it's amazing. It's not that he just learned 1, 2, or 3 words, but these are whole sentences. And producing them in a sequence that makes sense. About 75% of the time, he'll produce words in a random order or a seemingly random order that don't make sense to us. And about 30% of the time, producing them in appropriate context with appropriate conversation. And he learned 400 words by the time he was 4 years old. That's remarkable. And so every once in a while, a study comes out on a non-human primate where they're saying a great ape, in this case, a gorilla, who's lived with humans for 39 years, is learning how to do human speech, or just speech in general, or learn vocalizations. And reporters call me up and ask me, what do I think, compared to these parrots and so forth. And here's an example.
How about when you're coughing? That was good. You did a sneeze and then a cough. Excellent.
Now, this is Coco, a gorilla, and that's the best Coco can do. All right? What's going on here is that there's good auditory learning because Coco understood the question in English pretty quickly. There's decent voluntary control of the limbs here, coughing like this, and even voluntary control of pushing air through the vocal tract. But the actual muscles of the larynx, Coco cannot control that well to modulate that air going through the vocal tract. And that's what is different here. And so these kinds of comparisons and differences between species have led to scientists like myself and others trying really hard to get our closest relatives to say something as simple as apple, where these parrots, 300 million years removed from a common ancestor, can go even one further and say, golden delicious, dummy. What's going on here? And so why can we do these more complex behaviors that our closest relatives cannot? And this is the question that I and many other scientists have been asking. And there have been various theories as to why. And from an anatomical perspective, we've recently come up with a new theory called the motor theory of vocal learning origin that I'm going to describe to you next. But it's not the only one that's out there. There have been a lot of theories and hypotheses of what makes us humans different from these other animals. And some of these have become assumptions actually without enough evidence. One is the presence or absence of a forebrain area that controls the vocal muscles that's present in humans and songbirds and parrots, but not monkeys. Right? Another is there is a vocal motor area here in the forebrain, but in humans and these songbirds, there's a connection from the auditory cortex to the motor cortex that doesn't exist in the other species. The other is that there's a direct connection from the, I said that backwards. That's the one I was just saying. Here, this is a connection from the motor cortex of vocalizations to the motor neurons that control the voice. The other, coming out of the linguistic community, is that there is a separate language module in the brain. This is the Chomsky hypothesis, that interacts with auditory hearing information and vocal information. And that language module only exists in humans, whereas the other species do have these auditory and speech-like areas. Or there's a general difference in the circuitry or the number of neurons, the brain size, in those that can imitate and those that can't. Or there's actually a difference not necessarily in the brain, but in the organs themselves. In other words, the idea that in humans, the larynx is more descended so we can produce more sounds compared to a non-human primate. So all these hypotheses are out there. Which one is true? My work in this area began when we found that the act of singing in songbirds is associated with the rapid increase, here in this case, is a song. That singing behavior is associated with a rapid increase of what we call messenger RNA for particular genes in the brain, in this nucleus called HVC in area X here, in the bird brain, such that for every song bout a bird sings in a half-hour period, we see this white signal here, a gene product turn on, like an MRI signal, right? And so what you see here is the front of the brain, this is the back, the cerebellum, the forebrain here. And an animal is remaining silent or moving around, you don't see that activation. Lots of labs have been studying the mechanism of how these genes are regulated in the brain during behavior and neural activity. We've done that as well, but we've also used it like an MRI signal to map communication circuits across various species of birds. And what we found is that all species of birds that we've looked at, colored in blue, have this auditory pathway in their forebrain, consistent with the idea that all of them have auditory learning. But only the vocal learners had what I colored red and yellow here, forebrain structures that were active during vocalization. And best studied in songbird, the red structures here are responsible for the imitation, and the yellow structures here are responsible for producing the imitated sounds. And we couldn't find anything like that in a chicken or a pigeon brain. So either, and what was remarkable is that there are 7 structures in the forebrain of each of the vocal learning bird group with similar connectivity that seem to have evolved independently at least 3 times, which is quite remarkable. Actually, when we first published this, some religious groups got a hold of this, put it into religious texts to say that this helps prove that there is an intelligent designer, because how could this happen multiple times like this? So that's one hypothesis, right? Or maybe there was a common ancestor back here and there are multiple losses in these open circles here. Or maybe everybody has it to various degrees. We just, maybe 1 or 2 neurons we can't detect in the pigeon brain. Or I didn't appreciate at the time, I'm a neuroscientist, maybe the phylogeneticist got the tree wrong, okay? And these are all really close cousins to each other. And there's one common origin. Right? So what is true? I don't think the tree could be wrong when you compare it to humans. That is, if birds can come up with a similar solution to this ability to imitate sounds the last 65 million years, are we humans any different in our brain anatomy from them? And so began to make that comparison. The first lesson you learn here, this is a songbird or a zebrafinch brain. This is a zebrafinch brain to scale with a human brain. You can fit roughly 3,000 of these into one human brain. Size doesn't matter. Right? Here is cortical folding, smooth cortex here. Cortical folding also doesn't matter for this trait. What I argue is the presence or absence of a type of neural network that controls the muscles. That matters. But to get at comparing the neural network in birds with humans, we had to actually deal with an all-dogmatic view that has been around for over 100 years about the vertebrate brain. And that view, beginning with in the late 1900s by Lewin Edinger, argued, taking Darwin's view of evolution, Aristotle's view of scala natura, and arguing that evolution was linear, not a tree. All right? And the goal of evolution was to generate mankind, particularly Europeans, actually. You'll see in this hypothesis. Where the idea here was that in the human brain, you have these basal structures they call basal ganglia, named them by old names, paleostriatum, that was there in fish, and then passed it on to amphibians who developed the archistriatum, who passed that on to reptiles and got a neostriatum for new in reptiles, and so on in birds, all these basal ganglia structures. And this is thought to be the cortex here, very tiny little cortex involved in just olfaction or something like that. And so I organized a consortium. Oh, by the way, further, it's thought that the neocortex, it's called new because it's only thought to be in mammals, and that it's biggest in Europeans, next biggest in Asians, and then smaller in Africans, and then monkeys after that. And these are the texts that I'm reading back in the 100 years ago that came up with the term neocortex. That's what it comes from. All right. And so I organized a consortium of scientists in the early 2000s to evaluate all this evidence because we knew something didn't make sense about it. And using the literature, doing our own experiments, we came up with this now we call a Martin view, where the bird brain actually has a large cortical territory, not just a small one. It's as big as it is in terms of relative brain size to the human. And here's the true basal ganglia structures. The difference is that in human brain, these are layered sets of cells, and these are clusters. But the cell types are similar. And so with this new view, we can now compare bird brains to human brains. Oh, by the way, when we published this, some reporters got a hold of it and said bird brain should no longer mean stupid. All right. So putting that together, I argue that we humans have inherited an auditory Wernicke's area involved in speech perception from the common ancestor with these birds, that it's homologous to these birds. This is why dogs can understand sit, siente say. But we humans have independently evolved structures in our forebrain and other brain regions that are functionally analogous to these regions of song-learning birds that you cannot find in our closest relatives. You cannot find this Broca's area functioning like the way it does in humans, in non-human primates. How could this come about? And I even argued that there's cell type similarities in these brain regions to the human brain between these birds and us. So, by the way, when this got published, this also was used in some religious texts to support intelligent design, because now 300 million years of separation from the common ancestor and something similar. So what is going on here? I didn't really have an answer until an accidental discovery where we found that not only the song-learning nuclei show this gene activation with singing, but the neurons immediately surrounding them show gene activation with movement behavior. In this case, we think learned movement, hopping in a rotating wheel for the first time. And like the song-learning nuclei themselves, the activation is independent of auditory input, is independent of visual input, is independent of somatosensory input, that is touch. That is, it's motor-driven. It's a motor pathway. And the connectivity of these regions around these song nuclei is similar to the connectivity of the song-learning nuclei themselves, with some differences. This makes a direct projection to the neurons that control the voice. This makes indirect projections in the brainstem to other neurons that control other muscles. So that was interesting. Further, we found that all vocal learners had this pattern, And the species that did not learn how to imitate vocalizations, they still had these motor circuits, but they did not have the song learning nuclei inside of them. So around the same time, another colleague of mine discovered that if he puts dancers, this is coming back to my dance life, into an imaging instrument and gets them to do choreographed sequences of movements, the areas of the brain that show highest activation in our brains are ones that are directly adjacent to laryngomotor cortex here and Broca's area, the speech areas. And so what was further interesting around this time was shown that only vocal learners can learn how to dance. And here's an example of a parrot, in this case a cuckatoo,
So
who can
I
learn how
read
to
it.
synchronize his body
this
to
is so much better than
musical
Dancing with the
sounds.
Stars. Tell us very briefly what Snowball can do, and then we'll witness it for ourselves.
Well, Snowball can dance to a beat, and he actually adjusts his dancing to fit the tempo of music.
Shall
So he
we?
does this in response to music, seeing somebody else do it, but he is listening to the music. And this is particularly dancing on the down.
Does he like for
which is hard
everybody to
other
join
animals
in?
to do. Here we go.
Shall we?
And here are some scientists, and I want you to compare the scientists to the bird. So you can go look that up. I don't have time to show the whole thing, but it's remarkable. You can't get a dog to respond. Dog will respond to music, but will not synchronize the whole body to the rhythm of the music. So what's going on here? All of this led to what I call the motor theory of vocal learning origins hypothesis, and that is I argue that all species that vocalize have this brainstem structures that control the innate barking of a dog or the chicken crowing and so forth. All of them have a forebrain circuit here that controls learning how to walk, learning how to fly, learning how to do gesturing in non-human primates, even sign language gesturing. I argue that during embryonic development, this motor learning pathway is replicated multiple times to get hooked up to different muscle groups to control different motor learning abilities. But that in vocal learners, I argue that this motor learning circuit, through some kind of genetic mutation in us and birds, mutated and created a new vocal learning pathway, replicated one more time, took over control of this innate brainstem structure to then get this emergent vocal learning pathway that's similar in us, in the different birds, and possibly other vocal learners that have been looked at yet. So if this were true, it would mean that the genes that function inside these brain circuits for vocalization should be similar to the genes that function outside of them. The genes that function outside of them should be similar across all species that have these motor learning circuits. And there should be some differences that are associated with the difference of the connectivity inside the vocal learning areas and outside of them. So this is the third part of my talk, where we began to try to test this hypothesis, which is a difficult one to test if you don't have the genes of all these different species. So I got involved in sequencing the genome of one songbird, a zebrafinch, and a parrot here. And that led to a number of papers from various labs, including my own. But you begin to realize, for any of those that know statistics, if you have an N of 2 vocal learners and one non-learner, a chicken sequence at that time, that's not enough to find the genes associated with a particular trait. You need more than that. Plus, a chicken is as far removed from a songbird and parrot as marsupials or mammals are to us. We need a close relative of these songbirds, like we need a chimp, for example. And so how are we going to do that? I got involved with what's called the Genome 10K Consortium, whose mission was to sequence the genomes of 10,000 vertebrate species, but they didn't have any money. They partnered me up with Goji Zhang and Tom Gilbert of BGI and University of Copenhagen, who also had interested in birds, whether the tree was right or not. And the idea is if we had whole genomes, we can use that information to find out if the tree that we were looking at was correct. So we convinced the leadership at the Beijing Genome Institute, brought a lot of collaborators together, and sequenced the genomes of 45 new bird species, representing all bird orders across the family tree, including multiple vocal learners of songbirds, parrots, hummingbirds, and species that were considered close relatives, but according to different publications. So one year, we see a paper out there saying falcons are the closest relative of parrots. And another year, somebody says, no, you're wrong. It's owls are the closest relative of parrots. It's like one year finding out chimps are our closest relatives, and another year finding out, no, it's a gorilla. All right, who are you going to use as a control species to compare with for language abilities. So what we did is just sequence them all according to the different publications. It wasn't cheap, but the people we convinced at the time were really convinced this was a good idea. So when you convince people it's a really good idea, somehow money shows up. But also a lot of people showed up. All right, so we asked people to join us in collaborations, but a lot of people also flocked to us because with all these genomes, that had never existed before across the vertebrate lineage. So by the end of 4 years of toiling away at this data, we had over 200 researchers from 110 institutions here from 20 countries looking at this. And in 2014, we ended up publishing 28 papers from this effort on one day. And between 2014 and 16, 50 papers altogether, of which 8 of those were in a special issue of Science magazine. So you can see what citizen science can do. I don't want to take credit for all of those papers. So in red, I just color-coded those in which my lab led, mostly in neuroscience, and in blue, in which those studies that my lab contributed to, and in black are papers in which these folks benefited from these genomes, but we didn't contribute to intellectually. And of course, I'm not going to tell you about all these papers today, but the most relevant ones, I'll just say a few things about. One is, using these genomes, we were able to create a genome-scale vertebrate tree of all birds here, all bird orders. And what we do is compare it to this tree I had been using for over a decade of comparing vocal learning species. Look at all the rearrangements in the tree when you use the whole genome. Actually, this tree is mostly based upon body morphology, and it's most similar to protein coding genes that are rapidly evolving, which is very interesting. It's a convergent tree. What about the vocal learners in the red here? They're close together in this tree, where in this tree it pulls hummingbirds much further away from songbirds and parrots. So it really does indicate independent evolution, at least 3 times, if not 2 times, followed by several losses. If this were true, it would be like humans and chimps having a common ancestor that had speech. Chimpanzees losing the ability to speak and humans maintaining it. So all these hypotheses are quite remarkable. What about the brain areas themselves? Now with these genomes, we can compare different species. So what we did is laser dissected out the song learning nuclei of all the vocal learning bird groups and their surrounding brain structures, profiled them to what we call microarrays that have all these different genes on them. And we did that for vocal non-learning species as well. And with the Allen Brain Institute was also had 3,700 samples they got from 6 people, postmortem brains, and several hundred samples from 6 macaques. These are people who donated their brains to science, by the way, okay? From different monkeys, and also profiled them to what we call these gene arrays. To look for the activity of thousands of genes and across all these brain regions in humans, these birds, and these monkeys. And the students in my lab, who now have their own labs, analyzed this data and generated some of this data. We came up with 7,000 genes that are expressed in the birds and human brains and used new computational algorithms we developed to find that roughly 50 to 70 genes out of these 7,000 showed similar activity in songbirds and humans that was not there in non-human primates or chickens, right? And what was that like? This nucleus that produces learned song in songbirds show a gene or molecular profile similar to human laryngomotor cortex that controls speech production. This nucleus also showed similar to another cell type there in terms of its molecular profile. And same thing for Broca's area, same thing for a part of the basal ganglia here. That is, these regions here are more similar to structures in the human brain in their genetic profile than they are to a chicken. All right? How could that happen? By the way, this again was picked up by religious groups. Said this has got to be intelligent design or something's going on here. And I don't have an answer as to why that happened. Multiple genes converging for speech and song in us and these birds. And so we did not find that, like I said, these regions in non-vocal learning birds or non-human primates. And just to show you the raw data for some of these genes, the axon guidance genes here that control connections in the brain, are downregulated or shut off. And you don't see that in the non-vocal learners. You do see them turn down, the white and the blue signal here, in the human brain. What's interesting about humans and parrots is we found 2 regions that show this differential gene activation. All right? And that led us to a new hypothesis where we argued that there's this ancient motor learning pathway, learning how to walk, learning how to fly, learning how to do all these non-vocal behaviors. And out of that brain pathway, by brain pathway duplication, emerges this vocal learning system in red here, in songbirds, parrots, and humans. But in parrots and humans further, we argued that this vocal learning circuit duplicated one more time in yellow here and created a new vocal learning circuit surrounding the old one to generate something that's more complex in parrots and humans. And in parrots, this new, what we call shell song system, is bigger in species that learn how to imitate human speech more readily than others. So, so a new hypothesis we call brain pathway by evolution by brain pathway duplication. So what's going on here? Why turn down some of these genes? Well, this gene SLIT1 is a gene that when it binds to its receptor called ROBO1, it repels the connection from forming. And by the way, these two genes, when mutated in humans, causes some autistic and speech disorder deficits. So we think it's involved. So why repel this connection from this neuron forming here? Whenever you have a direct connection from the cortex to the neurons that control the muscles, you get more fine motor control. So we think what happened in humans and songbirds is some mutation turned this gene off in these cells so you can make this connection here to have more fine motor control of the voice. We'd like to actually experimentally test that. How would we do that? We would try to upregulate the gene in songbirds. We can't do it in a human for now. See if we can break the connection, make it indirect, and prevent vocal learning. Conversely, downregulate it in a non-vocal learning bird, make a direct connection, maybe even take over an existing motor learning pathway and get the chicken to imitate, all right? And maybe even try that in a mouse. And I knew when we were doing this, we would one day try to get to mice, but at the time we were doing all this, everybody was assuming a mouse is a non-vocal learner, it can't imitate. And then a colleague of mine, Tim Hawley, came along and showed that mice produce these, this is a sonogram of sound here, ultrasonic songs when pitched out to our hearing range sound like this. That sounds like a songbird. And here's a male chasing a female, trying to court her with his song. Basically, they're using it for a similar principle as songbirds do. It turns out that our lab in numbers has studied this behavior. There is some level of plasticity, some level to modify their vocalizations in mice, but it's not like a songbird or a human. Further, if we look at the neural connectivity of the circuit controlling this, surprisingly, we found the region in their forebrain that has these neurons here that connect to the neurons that control the voice, only thought to exist in humans and songbirds before this study. But in mice, it's very sparse, 1 or 2 connections per cell here, as opposed to hundreds to thousands in humans and songbirds. So what about the genes? The genes that were downregulated in this speech area in us humans and these birds was not downregulated here in the mouse brain. So what we'd like to try to do now is actually downregulate it in the mouse brain, make it more like a human genetic pattern, and see what happens to these mice vocalizations. Fortunately, what was good for us is mice changed its vocal behavior. But instead of vocalizing something more complex and more, these mice vocalize less. So we don't know what's going on there. Maybe we have to manipulate all 50 genes. And that's what we're trying to head to next. Or maybe a master regulator of all these 50 genes that are specialized in these human speech areas. But what's interesting about this gene is that it is regulated by another one called FOXP2. And some of you may have heard about this gene. It's the first gene discovered in humans that, when mutated, causes a speech deficit. It's a gene that regulates other genes. And here's a child with a mutation of this gene.
Your name?
Laura.
She's trying to say Laura.
Where do you live, Laura?
She's trying to say Sheffield.
And how old are you?
4.
She said 4. So this is a 4-year-old child who has good auditory learning skills, understands human speech, is able to answer quickly, but has difficulty sequencing the syllables, the phonemes, to make good, complete learned speech sounds. So that prompted my lab and a few other labs to look at this gene in songbirds, and we find that here the white signal is where the gene is upregulated in the brain. It's called the basal ganglia, as I said before. It's upregulated in the area X song learning nucleus when a young juvenile bird is learning how to imitate song from an adult, and turned down when that learning period is over. By the way, what makes this such a nice model to study is that in us, it takes us 13 years to go through puberty and crystallize our learned song or speech. These birds do it in 90 days, all right? So this is its crystallized or adult period after 90 days old. So a colleague of mine, Constance Scharf, in her lab, they took an inhibitor of this gene, hooked up to a green fluorescent protein, knocked the protein down here, and asked, can these juvenile birds learn how to sing, all right? So here is a control experiment, a sonogram. This is a zebrafinch song. It's not as quite nice as a canary. Here is a control animal who has some other gene knocked down, not this one. This is what's supposed to happen. Thank you for tuning up. Here is another animal as a tutor. And here is his 2T with FOXP2 knocked down. I think you can see and hear that he did not make a good, accurate copy of this song. He got the acoustic structure wrong, but the sequence is semi-okay. So the same gene is being used for vocal learning in humans and songbirds, separated by a common ancestor of 300 million years from us, that didn't have this behavior. But then again, we asked, what about mice? We said they have this rudimentary connectivity and behavior. So working with Simon Fisher, we had generated mice with the KE family mutation, the human mutation, and asked what happens to their ultrasonic songs. What we found is that if we use the same tools that we use in songbirds, we found that these mice in black bars here, when you put them from a context of just they like to sing in response to female urine, put them in a presence of a live female, the males, they switch from this more simple song to this more complex song here. All right? But if you introduce this FOXP2 mutation, like humans, these mice can't produce these more complex syllables as easily. So even though it's mostly an innate behavior, even before speech evolved, this gene is being used for sequencing of vocalizations, of more complex vocalizations. So by the way, the females, if you play these more complex versus these more simple songs from two speakers here, the females spend more time listening to these more sexier songs. All right. So what about the neurons that are connected, I said, that have that sparse connection to the vocal organs in the mice? They're there in the mice that have the FOXP2 mutation, but unlike the wild-type mice that have those neurons localized in an area of the brain, the FOXP2 mice have these neurons spread out more in the cortex. So something about this gene is controlling the position of these neurons that modulate vocalizations. So this means that it suggests we might not have to introduce or induce a novel vocal learning system in the mouse brain, take a rudimentary pre-existing system and enhance it. And maybe we can get the mouse to, I don't know, say something. So, so to end here on this part of the talk, I'm going to summarize what we've learned of these various hypotheses that have been out there. I didn't have time to show everything because I want to get into my journey here. But I'm going to say that presence or absence of a forebrain pathway, we're also saying enhance forebrain pathway that's already there. More of a continuum hypothesis of vocal learning. It's not black or white, but there are a lot of shades of gray in between. Enhanced direct connection for the brain from the cortex and the neurons that control the voice. We don't find evidence of this direct connection between auditory cortex and vocal motor cortex that's different. We don't find evidence of a language module that's separate from a speech or spoken language circuit. We do find some evidence that there is a general circuit difference or number of neurons. Birds, especially vocal learning birds, have more neurons compacted in a small space than many mammals do. So a parrot brain has just as many neurons as a monkey brain, who's 5 times its size. Right? So that difference exists, and I think that might handle the extra brain space for these vocal learning systems. There is no difference, that, as far as we can tell, consistent difference in the vocal organ between vocal learners and non-learners. So we're getting closer to understanding this difference and maybe one day getting these guys to be able to talk to us. And that's my long-term mission, and for students and postdocs that want to come join us, this is what we'll be working on. So the personal journey part. I've thought about this a lot. I get asked questions about my personal journey a lot. And actually, it really has influenced the science that I've done that I just talked about for the last 45 minutes here. And so in the last 15 minutes, I'm going to finish up here to say what that's about. My science journey. I began my science journey as a dancer, all right, at the High School of Performing Arts in New York, where I really took what I've learned there and applied it to science. And when I graduated there, I decided I wanted to become a scientist. I went to Hunter College. I didn't have really any good opportunities. So the MARC and MBRS programs from NIH gave me those opportunities, where I stayed in the same lab for 4 years with Rivka Rudner, worked really hard like I did as a dancer to do good science in bacterial genetics, and ended up publishing 7 papers out of that work, 2 first author ones. Eventually, when I was a graduate student, some of these were published. That made me competitive for good graduate schools. I went to the Rockefeller University, stayed there for 6.5 years as a graduate student, not even stay there as a postdoc in the same lab. And I'll tell you, I broke some rules here. And I'll just tell you right away. The rule that I use is, what are people going to remember me for when I die, after I'm gone? Okay? Is it whether or not I follow the rules of staying in the same lab, a different lab, following, write your grant this way and that way? No, it's by the scientific discoveries I make. And however I make them can be a different path for different people. So choose a path that leads you to a specific discovery and not following specific rules that sometimes don't apply. So from Rockefeller, I went to Duke University as an assistant professor. In my first 6 years, we published 17 papers. Now that's with students and postdocs, of course. Became tenured, and then in 10 years published 80 papers in that time period. Full professor in 2016, and after that, then I left and went back to Rockefeller and published 7 papers since then. So this, it almost sounds like a show-off slide, but I present it in this way because when I win some awards or publish some papers that get some recognition, I get people asking me, how did you do it? I said, did what? How a kid from Harlem, New York, African-American, growing up in a poor neighborhood, do this? I says, I never knew I couldn't, right? And so even getting some of these awards, like the Alan T. Waterman Award from NSF that you heard, Pioneer Award, Howard Hughes position, and finally, recently I got an Ernest Everett Just Award. Many of you know that he was an African-American scientist who had a very difficult time. They wouldn't give him a job at Rockefeller. So he went to Europe. And so I'm getting an award in his name and asked to write an article about how did I go through my journey. And so I wrote this article, Surviving as an Underrepresented Minority Scientist in a Majority Environment, because I was tired of answering the question, how did I do it? So I figured, let me write about it so a lot of people can read it. But I'm here to tell you some of the major pointers for me. And there will be 4 pointers. One is not even about being an underrepresented minority, but just being an artist. Being trained as a dancer actually did train me as a scientist. Here's when I was in high school at the Westchester Ballet Company in the Nutcracker. I do have to say Ethnicity does even play some role in here because I was the Arabian guy in the Nutcracker, and you can imagine why. All right. So, and just one point here is that the transition between science or dance and science was natural for me, as both required discipline, creativity, hard work, often acceptance of failure before something works. And so really the two kinds of fields are merged for me. Second point, role models. I really did have some good role models in my own family and outside my family. My family was my grandfather, not my father in particular. My father, who was more like a good friend to me, but he was going to college, he dropped out, he used what he learned in chemistry to start synthesizing drugs, sold them on the street, got involved in drugs himself, eventually became homeless, and so forth. And eventually actually was killed by teenagers who were trying to kill homeless people as initiation into their gang. And so, no, that wasn't a path that I had wanted to follow, but it's a path that I could have if, without knowing. But my grandfather was a guy who considered you need to be a person of excellence. And so I followed that track. Another role model for me was not in my own family, but was my undergraduate advisor, Rivka Rudner. She considered me her science Jewish mom. She really took me under her wing, held my hand, made sure I did well in her laboratory, and we worked together side by side, to publish those papers. So the role model doesn't have to be your ethnicity nor your gender. Opportunities is the third thing that I've really learned about. And what I've learned is that what I consider the equation of success is hard work, talent, and opportunity. And when one of them is missing, there's a problem. Many of us are in control, more in control of how hard we work, or even to a certain degree how much talent we might have. But opportunities is something that we're not that easily in control of. And what I've learned about opportunities is you have to take advantage of them. And if you don't, you could be missing out on this equation of success. But sometimes opportunities have to be there. And what I've learned about that is an example. When I was applying for faculty positions, I got offers from Duke University and other places, and including UNC down there in North Carolina. And I was trying to decide, okay, which one I'm going to take. And what was quite striking for me, the first thing was that these places, Julie was there at the time. I don't know if I'm seeing the same perspective, but I felt like I was being competed over like a basketball player, right? Because here is a person of color who's had the success which is unusual for them. That's one. And the second was, in terms of opportunities, was that Duke, they said, if you come here and you're a faculty member for 5 years or longer, then we will pay for your children's college tuition wherever they go in the world. If they go to Duke, it's full tuition. If they go somewhere else, it's 75% of Duke's tuition level. And Duke is expensive tuition. And this was amazing to me. This exists? Such a really? You can pay for my kids' tuition? I was like, I never heard of such a thing. And Duke, my parents could not be students or professors at Duke before 1968 because people of color were not allowed, right? So I could not benefit from this program, but generations of white Americans could for decades. So I realized that was an affirmative action program for white Americans, all right, that I didn't have an advantage of. And I'm benefiting from these affirmative action programs from NIH, sometimes internalizing, do I really deserve this? Is it unfair? No. These affirmative action programs that I benefit offset disadvantages that I didn't even realize I had. By the way, I did have 2 children, and now they've gone to college and they got their tuition paid for. So therefore, Duke had an affirmative action for me. So, so take advantage of those opportunities wherever they may lie. And the fourth item here is diversity. Diversity has been shown over and over again, including now more recently scientifically, to breed success. And in my lab at Duke and now at Rockefeller, I've had people from all walks of life, from all places around the planet, and gender and so forth. Bringing these people together with diverse views is like bringing physics and scientists together, biologists together, that led to the discovery of DNA, right? You have new ideas, and then if it was just monoethnic or monogender. Same thing for the actual scientific approaches. So I use all these different kind of approaches to answer my question. And how did this come about? This just happens naturally for me. But I started digging into my own psyche and realizing it's how I grew up. I grew up in a family that was active in the civil rights, and we had to decide, or we were arguing over, are we a Martin Luther King family or a Malcolm X family, all right? Most of my family were Martin Luther King supporters. Love everybody. Bring everybody together, and then you'll get a greater good. Bring all techniques together. I was practicing Martin Luther King's approach in the research in my lab. But I still did listen to Malcolm X a little bit, and by any means necessary, okay? Learn a technique, bring a collaborator in, use any means except for killing somebody to figure out how to solve the scientific problem. And those are the kind of thoughts that gone on through my head back then and still till today. And even in my own family, we were arguing who we are. Some saying you're not proud of your blackness, you're not wearing your hair like an afro. Or some would say you're denying your European ancestry, or Native American ancestry. So when I was a graduate student, and from there on even traveling back to North Carolina, where a lot of my family is from, I did a lot of oral history, and this is what I've been told, for example, in the last few generations. Several African, Native American, and European groups. And so I actually started to do this genetic analysis, sequence my own genome. Well, here I'll show, but even before I get to that, I had my own genome sequenced by David Goldstein's lab at Rockefeller, at Duke, when he was there. And then Carlos Buscamonte's lab at Stanford, they analyzed it, because they really wanted to see if all this was true. They were helping me out. And the first email that I got from Carlos's lab was from a postdoc of his, is that I was 77% European, right? I said, no way. Who am I representing here, right? And so I sent a bunch of emails of all the darkest people in my family and my darkest ancestors, right? Says, this can't be true. And so a few days later, that's the morning I woke up white. That's what I say. Right? So a few days later, I get another email saying, oh, we used the wrong admixture database. We used the wrong database. We got to use an admixture. We didn't realize you were mixed, right? And so, so I came back 40-something, 43 or something % European, West African, and a tiny bit of Native American, which they had a hard time believing. But the Native American ancestry is not represented in these databases. So I've been working with Ancestry and 23andMe to try to do that. And hopefully that'll improve. But my Ancestry.com data says basically 7 African groups and 7 different European ancestries in my genome. And if you look at that with 23andMe data, the purple, different shades of purple are the different African ancestries, the different shades of blue are the different European ancestries, and here's a little bit of Native American here in orange. You can see it's all jumbled up. It's like, genomes going wild, basically. And which means there's been a lot of admixture in the last several hundred years in my family line. And this is also present in other African Americans. What about a person from Africa? This is what their genome looks like, okay? And same thing for Europe, it'd be all blue. There's actually been a lot more admixture going on here in the United States than there has been back in European and African countries. And so the 4 different versions of me that I grew up over time is that here's my perception, African American, here's society's perception of me, right? A little bit of European and Native American. Here's my oral history, 50% African, the rest Native American and European, and here's my genetic results, all right? I'm like Obama, basically. And so, and it really awakens you up to how connected you are to people and so forth, and this perception of what we are and this actual genetics of what we are can be very different. And even this might be wrong because of the wrong databases being used. So to finish off here, I'm going to add a few more points about my journey. I didn't have time to show the actual graphic slides, but they're points that I've learned along the way. I've been trained to persevere in the face of daunting obstacles. Been told sometimes you have to be not as good as, but better if you're going to succeed because of my ethnicity. And overcoming the feeling of less than, which I've had that feeling many times, I only overcame those feelings by being successful at publishing papers or getting grants. It's hard for someone to tell me that. My skin color or gender, I learned, is usually a disadvantage or advantage. It's rarely neutral, all right? And so what I've done is try to ignore that, but try to deal with these 2 jobs here, be the best scientist I can be, and also cure society's racial disease because of my ethnicity. I'm the one who's thought to have the solution if I get hired as a faculty member. I think everybody needs to be part of this solution, but what I've decided to focus on is to be the best scientist I can be because we need more people to lead by example. We need more role models, and that's going to change people's perceptions. When I went to Rockefeller University in New York, and as well as Duke, especially back in 1998, I felt culture shock because I felt like I was in a different world. I really did feel like I was in the middle of Europe. So I had to learn how to reach out to people that I'm not used to learning how to reach out to. That's another lesson that I've learned. And not everybody's, people will help you if you reach out to them. And the other is accept all approaches, Martin Luther King, and by any means necessary, Malcolm X. And so, yes, even more so importantly now, become active. Scientists are not taught to be active in the community. And so yes, I did talk at the Science March for Science. Was a little afraid to do so, but speak up, because we need that more now than ever before. And I'll just end here with the 3 different summaries. Say that vocal learning evolved multiple times using the same genes and similar brain circuits. Brain pathways for these complex behaviors may evolve by brain pathway duplications. And this whole hypothesis here could lead to a theoretical path to engineer such brain circuits or maybe their repair. And as I showed in the second half of the talk, our culture, ethnic background does influence the way we do our science and think, and it influences how I pursue understanding the brain and mechanisms of complex behavior. And I'll go ahead and stop there. Thank you. You're welcome.
So we have time for questions. There are microphones there.
On any topic.
So you know what? Instead of I always hate this. Just grab the microphone behind you and yank it out of the. Sorry, guys.
Or pass it
How
on.
much
Hold
of
it out so we can hand it around.
how much of
I'm gonna put some credits
has
up
an
first.
influence on, say, mating and reproduction, and then how might that impact the selection differences that you've seen?
How much does songbird name? Say it again?
How much of vocalization and song, especially in non-human organisms, have an impact on reproduction?
Oh, reproduction.
Might be a selective mechanism?
Yes. Yes, so the idea, so there's a whole theory out there that even, vocal learning in general could be sexually selected upon. And that is, a lot of these species, these learned vocalizations are used for mating. And and that includes humans, Jennifer Lopez singing, for example, right? Ricky Martin, and so forth. But in only some species, like humans and dolphins and parrots, are these vocalizations used for more semantic-like communication. So the question is, why is it not so common if it's being sexually selected upon? And the answer to that came to me when I was sitting out at Duke Gardens one time reading a paper. And I'm hearing a song sparrow sing from the top of the tree. And I pay attention to him. He's going like, he keeps doing that. And then 10 minutes later, he switches to another song. And I look up again. And then 10 minutes later, switches to another song. And here is this guy at the top of a tree advertising himself to the rest of the world, saying, look at me, look at me. I'm not even a bird, I'm looking at him, all right? And he's trying to get a female to look at him. I think predators are also recognizing that and snipping them off. And so you have to avoid predation, I think, to evolve that trait by sexual selection. And it turns out amongst mammals, vocal learners are at the top of the food chain, like humans, dolphins, elephants, or produce ultrasonic learned sounds that others can't hear, like bats. And in birds, in that family tree I showed, turns out the common ancestor of songbirds and parrots was an apex predator that's now extinct. Sorry, go ahead. Yes. I'll let you navigate.
Quick question about FOXP2. So it sounds like with FOXP2 that it's not unique to sort of the fine motor program that's happening with vocal learning, even in humans and songbirds, right?
That's right.
So is there any other similarity that, or even if it's not unique, is it uniquely for vocal learning in these species unless something is disrupted, or is there some other sort of motor similarity?
Good question, because when FOXP2 was discovered, or is discovered to be involved in speech, people called it the language gene. So far, we have not found any gene, maybe a slight gene duplication here and there, but we've not found any gene that's specific for language, spoken language, or song in these birds. Instead, what seems to have happened is that old genes are being retooled for new purposes. And that's the case for FOXP2. FOXP2 is actually there in all vertebrates. It's in crocodiles, right? It was in dinosaurs. Maybe there was a vocal learning dinosaur. But so what's happened, it seems, is that FOXP2 evolved certain changes in humans, maybe in songbirds, and now it's this new behavior, speech, depends more on the gene than other behaviors, such that when you have this mutation in this child, one copy is intact, the other is not. So the child has some detriments doing some behaviors, but not much. The child can still ride a bike, can still walk, run, and so forth. But the speech is more dependent on the gene than other behaviors. Does that help? Okay.
So in answer to your first question, you alluded to...
Can put the microphone
being
up?
recorded, so if you could do that.
So in your first answer, you alluded to the fact that the males were probably singing more than the females, in birds in particular.
Mm-hmm.
Do you see there are some like sexual dimorphisms in the wiring between male and female birds that
Yeah.
correspond to these?
Yeah, so this is all
auditory
interesting.
areas?
So since we all live in a temperate zone, here's like how your location and your culture influence you. Most people think that only males sing amongst songbirds and the females don't. The actual reality is that most species, both sexes sing. It's just that most species are near the equatorial regions. Actually, humans evolved near equatorial regions. And so the further you get away from the equator, the more higher probability females have lost the ability, right? And males maintained it. Although females select the males on how well they sing. In the equatorial regions where both males and females sing, there is still sexual selection. Some females choose mates of guys who can imitate them well or duet with them well. And if you're not a good duetter, forget you, all right? And so what seems to happen mechanistically, to answer your question, is that all songbirds are born, that is hatched, with a developing song learning circuit. But what happens in the females, it atrophies as they become an adult. But chickens and pigeons, they're not born with anything like this. So it's a loss of a trait for whatever reason to divide up the labor between the sexes in more difficult climates.
Because in reproduction in birds as well, I mean, the females typically show this recondrence and regression of their ovaries when they're in an out-of-breeding condition.
Yes.
And I wondered whether you saw similar plasticity in the brains when they're in a
Yes. Yeah, so Fernando Nottebohm and his students before my time at Rockefeller, they discovered that, yes, some of these, in the more temperate zone, they go through breeding periods in the spring, basically. Whereas in the equator, they breed all year long, like humans could. So in these more temperate zone where there's these seasonal changes, what happens is that in the bird brain, there's new neurons that are generated throughout the entire life. And so what happens is when they stop singing, they stop putting new neurons into these brain circuits and they start to shrink. And when the breeding season comes back, more new neurons enter and the brain regions expand throughout the year. So the brain is more plastic, or the ability to be more plastic is remarkable.
So I have a dumb question. The circuitry that you spoke about in terms of its duplication, I'm interested to know what ignited, or is there a known mechanism for basically the duplication itself? And is there any sort of hierarchical sort of circuitry mechanism for that?
Right. Glad you asked. And he's encouraged me to write my own book like his about between dance and biology as opposed to jazz and physics. But the circuitry, yes. So we don't know the mechanism. We don't even know if really the brain pathway is being duplicated. It's the best explanation we can come up with. But the hypothesis I'm working off now is that we know that phenotypic diversity, including in the brain, could be controlled by gene duplication. You duplicate a gene, the old copy maintains its old function, the new copy goes on to have a new function. So what we're looking for is to see if there's some kind of genes that have been duplicated more in humans and vocal learners that then cause a duplication of another motor learning pathway. Although you didn't ask this, I would want to say something about this from a physics point of view or from a circuit point of view, network, is what this suggests is that there are limited networks that the brain can generate to produce complex behaviors. And during evolution, the brain is just replicating those basic circuit motifs and modifying them in different ways for different behaviors. And spoken language basically is not something totally new, totally novel, brilliant new thing in humans. It's based upon an ancient motor learning pathway that's been amongst vertebrates for the last 300 or more million years.
I'm very sympathetic to that, and I think that was a wonderful talk. But in terms of the complement, I wonder whether you would speculate in terms of the differences between humans and birds one of the main things that sometimes people talk about is generativity, so the ability to sort of combine words in novel ways to express new things. I don't know that much about birds, and I suspect that there's some birds that do sort of just rote imitation or aren't able to sort of combine words to generate sentences when they want to convey something relatively new.
Right.
But there may be, sort of in parallel, there's also some incredible work in other kinds of birds in terms of their ability to sort of use tools and sequences in novel ways to accomplish remarkable things. I don't know whether there's any overlap in those kind of birds with the ones that are able to produce
I think,
vocalizations.
so the answer is yes and no in the sense that, well, Disco, the parakeet you heard earlier, is generative, can put human learned words into new combinations that are meaningful to us. You know, we can't ask him, did you say what you mean? He says yes, I don't know.
Right.
But in terms of non-vocal behaviors like tool use or some kind of sequential behavior, I think that sequential algorithms and network in the brain is already there before language evolved. That we're just overrating language, and that what language brain circuits have done is inherited that sequential type of syntactical-like structure of sequencing movements together for the larynx, in this case, from the surrounding motor circuits. And once it inherited, then it then went off and made it more advanced. And so yes, there's some birds that have tool use and so on. Now, what I didn't get to before, and your question reminds me, is that has something else happened in non-vocal behaviors after vocal learning evolved in humans and songbirds? And the answer is yes. So I think once you evolve this vocal learning circuit in integrating auditory input for the control of the muscles of the voice, that circuitry mechanism contaminated the rest of the brain and has allowed these vocal learners to learn how to dance. And that's something we're trying to test now, to see if there's some rapid auditory motor integration in the non-vocal areas. Second, for whatever reason, there's anecdotal studies showing that crows, dolphins, elephants, humans, parrots, seem to be the more intelligent species amongst their groups. And so the question is, has something else happened after vocal learning evolved that created more behavioral complexity? No one's really quantitated that or shown that mechanistically, but it's something that needs to be looked into.
Thank you.
Okay, anything else, especially from some of the students?
I think I'm holding
Before I
people
call
from
on
their
you,
dinner.
don't be shy.
Yes. or on the represent minority issues or anything like that.
Okay, well, I have a technical, stupid question.
Okay.
So, back in the day, I'm a dinosaur, so I used to stick electrodes into nerves and things like that. So this whole notion of looking at what's a gene being regulated as a indicator of behavior is kind of freaking me out.
Yeah.
So can you play with the mechanics of that and tell me what's going on?
So for a long time, before these genes were discovered, it was thought that the electrical signals in the brain are rapidly doing processing. All right, that's what's controlling your immediate perception and production of behavior. All right, and that the genes are kind of static. They get turned on, they stay there, protein product is made, it's stable, and whatever changes occur over days, if not hours, but definitely not minutes. What we've learned in one of the papers I didn't show you is that from this big genome project, is roughly 10% of the genome is dynamically being regulated. That is, gene products being turned on and off in the brain in different cell types, different categories of genes on a moment-to-moment basis throughout the entire day as behavior is being performed. It's a factory going on, basically, of genes turning on and off. And there are certain drugs that can affect these genes. So you can imagine that you can affect the dynamic activity of these genes in the brain and affect behavior. Why that's the case, I think it's two reasons. One is, as you use up brain activity, use up the proteins, you need to replace them so that you can talk a half hour later. If you don't replace them, your speech is going to start to deteriorate. That's one prediction. The other is some of these genes are being used to form memories of everything that's going on from a moment-to-moment basis and laying down those memories so you can keep them for a long time.
So it's activity-based as opposed to pattern-based?
Both. So certain patterns can cause certain genes to be regulated in certain ways, and just then ramping up the gain of activity within a pattern can change the level of activity of that gene. We do have more questions, all right. I mean, for those who want to go to dinner, go ahead.
This question is sort of back to something you said right at the beginning about how humans learn language much better before puberty. But children can learn different languages, right?
Yes.
And a child that is born to parents who speak one language can grow up and learn a different language.
Right.
Do we see the same thing in birds, where a young bird can be taught a different song than what its species song would normally be?
Yes, so that's what I meant by the critical periods, is that young birds, children, all right, learn how to imitate different songs, different languages, different dialects much easier. Okay. Now, amongst primates, there's only one vocal learner, humans. So you don't have anybody else to compare to amongst us. Amongst songbirds, there are 3,000 species of songbirds. And they're all thought to be vocal learners. And they have various degrees of vocal learning ability. So some are like humans. They learn best early in life, have a difficult time learning a language or even other things later in life, right? Some other birds, like the zebra finch, learns just one song and then can't even learn a new song later in life. And we think there are certain genes like BDNF, brain-derived neurotrophic factor, and others that are shutting off this critical period more strongly in a zebra finch compared to a canary, compared to a human, which is leading us to, we hypothesize, and I'm waiting for a student to do this experiment, is to take BDNF and some of these other genes and change its regulation in the adult, get the animal to learn a new language, shut it off so it can hold on to that memory, and maybe that'll be a mechanism to get us to learn new languages later in life easier.
Yeah. But I think more what I was asking is, if you take like a zebra finch and you put it as a baby with other birds, will it learn a song that's not a zebra finch song, but it's the song of a different bird?
Yes, so there's a genetic predisposition. If you take a zebra finch, put it in a cage next to a zebra finch and another cage next to a canary, it's going to preferably learn its own species song, all right, even though it has equal access. If you remove this other zebra finch and just give it the canary, it will learn how to sing the canary song.
That's pretty cool. Thank you.
And we think certain molecules like oxytocin and others might be involved in this, the social bonding with your own species.
Thank you.
Really cool question. I'm going to have to close this out, unfortunately, because we have to get him to dinner at Chris's house. So thank you so much, Erich, and thank you for coming to the first Thinking Out Loud this season.